Hello, it's a pleasure to be here. Thanks for the invitation. My name is Jose Manuel Baena, and I'm going to be talking today about creating functional tissues using bioprinting and bioreactors. A bit about me. I'm a PhD in biomedicine, University of Granada. I'm also an industrial engineer. I started working with 3D printing of medical device in 2008 and created my first company, Breca Healthcare, for manufacturing 3D printed medical devices. And then in 2011, I met a professor at the University of Granada, and he was doing autologous transplantation of chondrocytes and asked me for developing a 3D printer to print chondrocytes. We called the project Regemat 3D, and in 2015, after the development of the first version of the bioprinter, we started the company that is called Regemat 3D. Another project I am involved also Lab Methods inside Regemat, that is a community for the researchers to upload biofabrication methods, biofabrication protocols to help other researchers to advance and to speed up the development of solutions in the field. And also another more personal, I have a YouTube channel that is called Bioingeniería, that is showing to the students, to the community, the technologies and the new trends of bioengineering. Then I'm going to start with my talk very fast as an introduction. For me, 3D printing has three advantages in medical science. The first one is the ability to print, to create complex shapes. Anatomical shapes are very complex, and then we need a powerful technology to create these complex shapes. The second one is the ability to create a mesh structure. This is the only manufacturing technology that allow us to create a controlled mesh structure. This has advantage in medical device because we help the surrounding tissues to attach to the device, but also in bioprinting, because with the mesh structure we can mainly do two things. The first thing is we can control the biodegradation time, that is depending a lot on the surface, on the area of contact with the environment. And the second one, with a specific mesh structure, we can transfer loads to the cells, to the biological part that are the ones that are creating the cellular matrix to create a specific and a functional tissue. The last one, the third advantage, is as being an additive manufacturing technology, we have access to the shape during the procedure, and that can help us to introduce in bioprinting biological components as the cells. This is a bit my activity as engineer with Breca, creating the medical device. We started with titanium, the electrobeam melting, we moved to PEEK, to another polymers, and then we have been creating like hundreds of clinical cases, prostheses, implants, and a lot of solutions for surgical guys. Then I'm going to start talking about the first part of the procedure to create a tissue, that is bioprinting. Now it's very easy to explain to people what is bioprinting because a lot of people are working on bioprinting. For me, bioprinting is putting together 3D printing technologies, tissue engineering, more specific the part of the biomaterials, and regenerative medicine, the cells and the biological part. Then, but we have to know, to be aware that what we are creating with bioprinting is not a tissue. It's a 3D matrix, a scaffold with cells or without cells, but it's not a tissue. A tissue is a structure with cells, a structure with an extracellular matrix, and a structure with another component. Then, in order to create a functional tissue, we in the company have developed a procedure that is based on two different parts. Very important, both of them. The first part is the bioprinting. This is the manufacturing, the 3D printing of the biomaterials, of the bioinks with the cells. And the second part is the maturation. We have developed and we keep developing strategies and devices that can interact, that can apply stress, loads, stimuli to the scaffold with the cells to make the cells to create a specific cellular matrix and a functional tissue. And for us, both of them, both blocks of the diagram are very, very important. Then as application, we have different applications as could be biodegradable medical device. There are a lot of things to do with bioprinting, even without cells, printing scaffolds, printing matrix that biodegrade with biomaterials that have been already approved. There's a lot of things to do, and in the short term we are going to find a lot of applications in the clinical space. Then we have a second range of application that is the scaffold with the cells, that is useful also for an advanced therapy solution, but also to study, to know more, to learn how the cells interact in 3D, and also to do drug development using this 3D culture. Then we have another range of products that is the functional tissue, more in the mid and long term, and in the long term we will have organs, functional organs, and there are the four classifications of different products. In the company, we started with the pandemic. In 2020, we started a lab. I'm now the director of the biofabrication lab inside RegeMat, and we are working mainly in cartilage, in bone, in skin, and also in spinal cord. We are very happy and very open to collaborations. Then if you want to start a collaboration with the company, to apply for a grant, to apply for a project, just send me or send us an email, and we will be very, very happy to participate. Then, talking about the two blocks procedure, we have to develop a strategy to create a specific tissue, functional tissue, and we have to take into account the two parts of the block. The first part is going to be the biofabrication, the bioprinting procedure. Here we are going to choose the material, the scaffold geometry, the type of cells, the bioinks that we are going to use, and also another biomolecules, as for example the exosomes, the growth factors that we are going to apply. Then we are going to create a strategy to create this 3D scaffold with the cells, and after we are going to go for the second part of the procedure, and we are going to apply different stimuli in a bioreactor to make the cells to create a specific extracellular matrix that is going to be the tissue. And in our whole strategy for creating the tissue, we have to take into account both procedures. Then, more specific, and now I think around 80% of people working with bioprinting are focused on developing of biomaterials that first that can be regulated, that can be used for clinical application in the short term, another ones more for the long term. Then very, very important, the printability. We find a lot of material that can be printed easily, and another ones that are not very easy to print, as for example biomaterials that are derived from extracellular matrix. Then as engineers we want materials that can be printed and also that have a specific and relevant stiffness and mechanical behavior in order to print them, to apply stimuli. But we also have to take into account the biological, the cellular part, and cells normally are very happy with materials that are very close to extracellular matrix, but the printing procedure of this material is complicated. That is why we have to take into account the maturation procedure and the viability of proliferation on the cells, and to create a specific strategy. And in some cases, it's an indirect strategy. We will be talking about that afterwards. To choose a bioink, to select a biomaterials, printability, very important, the cell viability and proliferation, biodegradation time. This is something that is depending on the biomaterial structure, but also on the mesh, on the scaffold structure. We saw, for example, for materials as polylactic acid, that if you have a given volume, the biodegradation time can be very, very very slow, the biodegradation time of the volume. But if you create the same volume but with a mesh structure and you are increasing, you are multiplying a lot the area, the surface in contact with the part, and then you can speed up a lot and accelerate a lot the biodegradation time, even with the same biomaterial and with the same volume. Then, as I said before, materials that promote the cellular matrix formation, the cells normally are very, very happy with materials that are organic materials that are very similar to that they found in vivo, but normally we find a very specific, a lot of problems to print it. That is why we have to create a complete strategy, and then also the price and scalability. I mean, we found also, and we are working with several groups that are developing biomaterials that are very specific, that the cost of production is very, very high, and this is something that, or the prices are very, very high. It's something that we have also to take this into account, at least when, if we want to get to the final application with a mass production or with a application where we can cover a lot of problems from a lot of patients. This is the first block. Then we have been developing specific procedures where, for example, we found that there is, if you want to get faster to the clinical application, you need to work with materials that are already approved. Many of them are thermoplastics. The printability of thermoplastics is very good, but they normally use high temperature. And then we started to develop procedures that can inject the cells after printing a volume. This is very interesting because you don't have limitation on the material. You are just printing several layers and then injecting the cells inside. Also, a very important, interesting application that we found is that if you are using ultraviolet light for curing, for photopolymerization, you can print a volume, you can apply the curing, and then afterwards, when you have the solidified volume, you inject the cells. Then you can use injection volume filling for these two specific applications. And what we have been working is on developing strategies that you include a matrix and a skeleton, and after you fill it with extracellular matrix-derived biomaterial with the cells, and then you have the two advances. Then the engineers, we as engineers, we are happy because we have a structure that we can bring inside the bioreactor. We can apply loads that are going to be transferred to the extracellular matrix and to the cells inside the matrix. Then we as engineers, we are happy, and the cells are also happy because they are inside a material that the mechanical behavior is not very good, but we have the skeleton, but they proliferate and they have high viability. And as said here, the mechanobiology and activate the mechanobiology is key. It's a key for the cells to create a specific tissue. Then, in many cases, we found people that want to do it in the direct way, no? That is printing everything directly. But at the end of the day, what you want is the results. And there is a lot of opportunities creating indirect printing, as explaining before, no? It's printing the skeleton and after filling the skeleton with the cells. And there you can control, you can tune the biodegradation time, say before, and also this mechanical behavior that is what is going to, in the maturation procedure in the bioreactor, is what is going to make the cells to create a specific extracellular matrix. This was a publication, a picture in a publication that we did 4 or 5 years ago on the different strategies to print material that has been already approved, thermoplastic and non-thermoplastic. Then we did this trial. We used chondrocytes for the validation, and we printed the chondrocytes with alginate in a matrix PLA. This is quite old publication and project. And we show that after the printing, we can find high cell viability using electrovolume filling and also proliferation. Then we were also analyzing how the cells with laser scanning microscopy, how the chondrocytes are still alive after the product, and they keep proliferating on the scaffold. Then some example of application, we have been also working quite a lot on defining the best structure, the best scaffold structure for the cell, for the growth, keeping the stiffness that we want, keeping the mechanical behavior that we need in order to bring the scaffold to the bioreactor. Then there is still a lot of work to do in the part of the biomaterial. That is one of the main, nowadays, the one of the most important, and is improving the cell cultures. I mean, for everybody working on a lab, we know that there's still a lot of things to improve in the cell cultures, the biomaterials, and the biofabrication. We want to have biomaterial that the printability is high, but with good proliferation, with good cell viability. And then we have also several challenges in terms of the automatization, the regulatory, and say before, the price and scalability. Biomaterial is key. Here we have also some pictures on some example of the cells, then a matrix, in this specific case, also with thermoplastics and alginates to attach chondrocytes to the specific cell. This is what we have. Then after the cells move, the cells migrate. Then, in our opinion, to create the two-block strategy is very important, because to create something that directly you are replicating all the structure, all the very complex structure of a living tissue, this is going to be very complex. But the good news is that the cells, if you apply the right stimuli, the cells, they are living systems, they produce proteins, they produce a specific matrix, and this is what we are working. Then here also some examples, this for bone regeneration using biphasic calcium phosphate, and some research going and trying to show how the composition of the biomaterials, and comparing different biomaterials, we can improve a lot the biological response. And given that specific compositions, once we change the composition of a material, then the printing properties can change, and also the mechanical behavior. There's still a lot of things to do, but at least we are on the way of creating results, or creating data that help us to decide the best configuration for a specific application. Here we have another examples, or more on soft tissue, with using blending with alginate, gelatin, with hydrogels for the encapsulation of the cells, also with specific materials that can promote the formation of bone. We have, as RegeMat, we have a very big list of publications from our team, and also from our users. Then if you want to, we have classified by different tissues, by different biomaterials. We are very happy to share and to have you to focus your biofabrication project. And in many cases, this specific paper, this is on alginate printing. In general, we were using alginate as a bioink for containing the cells, but the printing procedure was very difficult. But there was a group just focusing on how to print very well the alginate, and we understood that if we dedicate time, even complex materials that show a relative good viability, and that are being used, or at least they are already with a lot of clinical data, different applications, that if we put resources on trying to improve the printing procedure of a material, we can do a lot of things, no? We can get very good results printing even very specific and very hydrogel-based biomaterial. Then I invite you to upload methods. I mean, we are in research, we're in science, the community is everything. I mean, we need the results from other people, and here you can share, and you can also get some advantage from understanding what are the components that you need in order to replicate, and in order to move forward the science and creating new results. Then we have to move to the second part, that, as I said before, this is very important. Again, and for me, it's very important to say that, being the director of one of the main players in bioprinting, what you print is not a tissue. What you print is a 3D matrix, a 3D scaffold with cells, but then the maturation procedure is is very important. Then we have developed, we are working already for a lot of years on bioreactors to apply stimuli to the scaffold after the printing. There is the BIM bioreactor we call bioreactors that mimic the anatomy and the physiology. That there are devices that you try to apply stress, uh, not like compression or blending. You apply the stress with the same shape that, in our opinion, in our hypothesis, has a lot of influence on the results. You use the anatomical shape in order to apply the stress, and also you mimic the physiology. You mimic the physiology, you mimic the CO2, you mimic the temperature, you create procedures. This is for us is a very interesting living lab where, without going in vivo or without using animals, we can monitor, we have a controlled environment, and we can apply the stimuli to the scaffold with the cells, and then we can play with different parameters in order to get the best results. We are working in different bioreactors, but the first one is one that is the knee bioreactor. We submitted a patent several years ago. That is one of the figures is shown here. This started several years ago. In 2018, we published our first publication on how the use of ultrasounds can apply stimuli to chondrocytes and make them to promote the formation of cellular matrix, and also as a quality control, you can assess how the cells are producing a specific cellular matrix. Then we have been in the last year working on the validation of our knee bioreactor, that is basically a knee. From a computer tomography, you get a knee shape, you implant the scaffold with different materials, you put in this specific result that I'm going to show you, quite preliminary. We were using the collagen type 1 from Porcine Origin, that is a brand that is from the German, that is called BioInk. We were using human mesenchymal stem cells from adipose tissue. In the next trial, we are going to be using mesenchymal stem cells from bone marrow, but as we had culture from adipose tissue from University of Granada, we're using it, and the skeleton and the matrix from polycaprolactone. Then we were applying the stimuli, that is compression, blending, different temperatures, and we were assessing the cell viability and the proliferation. With the confocal microscopy and a cross-section of the mitochondrial injury that we implanted at day 21, we did live death staining, as is shown here, in order to prove that the cells are alive. Of course, we are applying their mechanical stimuli and in a 3D matrix. And then this is, in some cases, you have a limitation on the frequency and also on the stress that you can apply, because you can kill the cells. And we saw something very, very interesting, that is the proliferation. With the dynamic here, the dynamic in gray is using the bioreactor, and the static is the same scaffold in a static culture. We saw that the proliferation is lower using the dynamic, and this is okay. We are doing mechanical stimulation, and we thought and we concluded that the cells are investing the energy on the cell metabolism, because, as I will be showing afterwards, we saw how the cells in the dynamic experiment were expressing the genes that are the ones, the markers for cartilage formation. And then we think that the difference could be because the greater investment in the metabolism in differentiation to chondrocytes. And in the application, appraising the compressive flow mechanical loads with this frequency, then with the physiological conditions, CO2, O2, temperature, we saw that it's very interesting, and that's why we are doing more experiments, and we are trying to amplify the range of, and also the days of the experiment. The activation, we saw the activation of genes that encode the transcription of factors as SOX9, collagen type 2, that is one of the main collagens found in cartilage tissue and also agar. There's very interesting results and promising results. We will keep working with this and trying to activate the mechanobiology of the scaffold with cells after the printing. And this is my talk. I'm just here. You can find me in social networks. Also send me an email. Also, if you're interested in starting a collaboration, just send me an email, a WhatsApp, and we'll be very, very happy. And also, if you understand Spanish, then you can subscribe to the Bioingeniería channel, and we will be also sharing different projects, results, also different technologies, and also inviting some different researchers in different areas that can help us all to increase the knowledge in the society of this amazing industry and technology range and science that is bioengineering. Thanks a lot. Muito obrigado.